Our first team of the afternoon and the seventh of the day is from New York City. I'd like to welcome Dina Malkina of HemoGenyx. Dina?
Thank you.
Thank you. You're on. Where in New York are you from?
Manhattan.
Manhattan. Where
Yes.
about in Manhattan?
Greenwich Village.
Greenwich Village. Oh,
Yes.
Marvelous. Great. So this was an easy trip for you.
It was short flight.
All right. Well, 10 minutes. You're on your own. Go kill it.
Thank you so much. Thank you very much for the opportunity to present today. We are delighted and honored to be here in Buffalo and to be a part of 43 North. I'll start by introducing our team. My name is Dina Malkina, and I'm the co-founder and CEO. I already have one successful startup exit under my belt, and prior to that, my background was launching new products for top technology firms ranging from startups to Fortune 500 companies. My co-founder, Dr. Vladislav Sandler, is our Chief Science Officer. He's a PhD with over 15 years of research experience at top hospitals, universities, and biotech companies with multiple patents filed. Our third co-founder is Alexis Sandler. She's an attorney specializing in intellectual property. In addition, we have a stellar board of advisors, including Dr. Jules Mitchell, who is the co-founder and president of a top CRO with over 300 clinical trials under its belt. And we're also lucky to have Ryan Bronze and Kartik Shah, both of whom have tremendous experience in leadership roles in their respective functions in biotech, medical device, and pharmaceutical companies. So what is the problem we're trying to solve? There are three key challenges that patients with blood cancers face today. First, there is a dearth of effective treatments for leukemia and lymphoma. Second, bone marrow transplants, which many of these patients require, have a very low rate of success today, with only about 50% of them succeeding. And then third, there's a critical shortage of donors, with about 70% of patients who need a bone marrow transplant not being able to get one. As you'll see in the forthcoming slides, there's a tremendous business opportunity associated with solving these challenges. But for us, there's also a very personal significance. My co-founder, Dr. Sandler, lost several of his close family members to these diseases, including his father, which was one of the main inspirations for his research and his motivation for finding a cure to these diseases. So our solution to this is replacing bone marrow transplants as they're done today with transplants of AHE cells. This is a special class of cells discovered by Dr. Sandler that reside in the liver of all mammals, including humans. When extracted, purified, and transplanted back to the patient, these cells give rise to blood stem cells, which are capable of regenerating the entire blood system that is cancer-free. The advantages of this therapy are threefold. One, because these cells are free of accumulated mutations, unlike other cells commonly used in these procedures today, the risk of relapse is minimized, and we expect about a 50% greater success rate compared to bone marrow transplants today. Two, the need for donors is eliminated entirely because the cells come from the patients themselves, so more people will be able to get treatment. And three, because the cells come from the patients themselves, there's a 100% donor match guaranteed, improving the success rate by 2x. There are other applications of these technologies also available, such as treatment for HIV, sepsis, multiple myeloma, and others. The market opportunity for this is large and growing. The current addressable market for leukemia and lymphoma is about $8 billion, growing at 7% a year. Additionally, by eliminating the need for donors, we'll further expand the market by $3 billion by opening the procedure up and making it accessible to patients who previously couldn't receive it. And then the additional applications that I mentioned will further expand our market by $30 billion to $41 billion addressable in 5 to 7 years. As you can see from our competitive landscape, HemoGenyx is uniquely positioned to succeed in this space. There's no other company currently on the market or in trials that plans to provide mutation-free, 100% donor-matched cells for bone marrow transplants. Although there are competing technologies emerging for treatments for leukemia and lymphoma, none of them can match our efficacy, and many of them are actually complementary. For instance, the T-cell CAR technology, in which for many patients actually still requires bone marrow transplants to complete the treatment successfully, meaning that they'll be able to leverage and benefit from our technology. The same thing is true for traditional treatments for leukemia and lymphoma, like chemotherapy or radiation. In many cases, those patients require bone marrow transplants as well. Our go-to-market plan for this centers around partnerships. We plan to and are already working with some of the top hospitals specializing in bone marrow transplants, which combined have most of the market share. For instance, we're already partnered with New York Presbyterian Hospital in this. Our plan is to involve these institutions in our clinical trials and leverage them to bring our therapy to market. The great thing for these hospitals is that they will be able to leverage their existing expertise to perform our procedure. And they will continue to make the same amount of profit, but they will bring a much more effective treatment to their patients. They'll be able to treat more patients because of individuals who previously couldn't find donors now having access to this treatment. And they'll be able to differentiate themselves from the competition. We'll also partner with other key companies, and then on the consumer side, we'll focus around awareness and educating the consumer on the availability and efficacy of our therapy. So why is Buffalo the optimal place to bring this go-to-market place to fruition? Well, there are several reasons. One, the Roswell Park Cancer Institute is one of the top institutions currently specializing in bone marrow transplants, and it's a crucial partner for us in order to be able to complete clinical trials and bring the product to market. Additionally, the new hematology and oncology plan by the Women's and Children's Hospital of Buffalo will be an important partner for us as well. Beyond those two institutions, there's also a rich research environment in Buffalo. For instance, Buffalo State University and the multiple medical device companies headquartered here and others. Beyond that, there are several well-known benefits of being in Buffalo also listed on the slide. Why is this good for Buffalo? What's in it for you guys? Well, one of the key things is having us here will establish Buffalo as one of the top treatment centers for blood diseases in the world. Because, as I mentioned, part of our go-to-market strategy is establishing those centers of excellence at institutions like Roswell Park Cancer Institute and the Women's and Children's Hospital. This will also establish Buffalo as a nexus for innovation in biotechnology. It will create jobs both internally within HemoGenyx as well as externally to support our operations. And because we'll establish our lab facilities here, we'll also be investing in the local economy and increasing the demand for infrastructure. As you can see from our business model, we have an accelerated path through clinical trials and FDA approval. This is because we're able to obtain an orphan drug exception for one of the applications of our technology. So we expect to do 200 procedures in the year after we're approved for leukemia and lymphoma and bring in $30 million in revenue that year, as well as become profitable in our first year after approval. Some of the benefits of our business model are, one, the flexibility to also apply our technology to other diseases, such as I mentioned before, HIV, sepsis, multiple myeloma, and others. The win-win for partners that I mentioned, the fact that hospitals will also be benefiting from this and will have a clear incentive to help us bring this to market. And also the opportunity for early exit. So because of our orphan drug exception, there's potentially an opportunity to exit before we ever hit our revenue milestones. There are numerous precedents of this in the market already, with some exiting for as soon as after 2 years for $60 million, such as Resolve Pharmaceuticals. Potential exit partners for us are pharmaceutical and biotech companies currently working on treatments for leukemia and lymphoma. such as Merck, Pfizer, and others. And with that, I'm going to leave up the slide, which will inform some of the questions that may come up in Q&A. And I'll invite my co-founder, Dr. Sandler, to the stage to participate.
Great, great job. Thank you. Really well done.
Thank you.
What'd you think? How was it?
I enjoyed it. I had fun.
Were you nervous?
I hope you guys did too. Not too much.
Not too much. Well, great. You did.
I've done this before.
You have? Oh, you did a great job.
Thank you.
All right, we're going to start with questions. David, we'll let you go first.
Thank you. You have been a part of the hematology group at Weill. I understand. How does this work relate to the ongoing work, which, from what I've heard, is quite similar at Cornell? And how is this differentiated from Dr. Rafi's work down there?
Right. I was the first author on the paper on reprogramming of endothelial cells into engraftable hematopoietic progenitors together with Dr. Rafi. And while doing that work, and that work was published in July in Nature, so while doing that work, I started looking at whether it's possible to find the cells that existed during development, so-called HemoGenyx endothelial cells in adults, and I found them. So this work was linked to the work I was doing for the last 6 years on reprogramming of somatic cells into hematopoietic progenitors. But on the other hand, this work is new, and this class of cells is a completely new class of cells that is being described now in the paper that I submitted.
Is the intellectual property settled?
Yes, the intellectual property is, I'm the only inventor on the patent on isolation and use of these cells. And the full patent application is being written right now by the patent office, by the Cornell patent office, and being filed. And we're negotiating the license deal.
Okay, so Cornell will be a participant in this deal? Is that
yes,
correct?
Absolutely, because the original patent belongs to Cornell.
Good.
We've also filed a couple of additional patents outside of Cornell this summer.
Yes.
I have some other questions that I'd like to give other people a chance here, Jordy.
We know that you have a lot of domain expertise here, David, so we're all happy to let you go interrogate a little further. Go right ahead. I knew all these questions, by the way, just so you know.
I would love to talk to you in detail.
Well, I would like to also. And I guess that raises a question in my mind. I'm not aware of any commitments yet, or any even introductions with Roswell or with the Children's Hospital. Are there some that I might not know about?
Not yet.
Okay.
But we are moving here, and we'll get into a discussion.
And you're going to be a very desirable partner.
Absolutely.
Okay. Where do you stand with the FDA? The federal FDA. In your work toward an IND for this.
So we have a great advisor, Jules Mitchell, Dr. Jules Mitchell, and Target Health, his company, who has about more than 300 clinical trials done. So he basically written up the plan of going to FDA and going through FDA for us already. So we haven't approached FDA because of different reasons yet.
Okay. And is there a second animal model plan besides the immunocompromised mouse model?
Yes, there are multiple experiments done in BL6, Black 6 mice already.
In what?
In non-immune compromised mice.
But what about larger animals?
We're planning to do it. We need some funds to do this.
Okay. That's it for the moment.
Thank you.
Just to add a little bit of color to that, and some of the experiments that Dr. Sandler has done so far on mice susceptible to radiation. In our control group, which did not receive the AHE cell treatment, 50% of the mice did not survive. And then in the experimental group, where the mice treated with radiation received their treatments, every single mouse survived.
That's correct.
Esther.
I'm curious how you market this, what needs to change in terms of delivery systems out when you get out into the clinical world. How long, again, is this survival for 3 months or for the rest of someone's lifetime? I mean, that's tautological, but how long does the effect seem to last?
That's a great question, and I'll start by giving you a high-level answer, and then I'm sure my colleague will have a lot to add. So the great thing about this therapy is, unlike transplants done today, these cells are completely clean and mutation-free. They look the same as they did as when the person was just born, an infant and a baby. So essentially, by using these cells for transplants, you're resetting everything to a clean slate. So you should have perpetual remission, and the risk of relapse is significantly reduced compared to current therapies. So, and the idea is that it's a cure, and the patient will survive perpetually until some other disease gets them.
In addition to this, I have an example of a cohort, a group of mice that survived for 11 months after transplantation. So in a normal mouse, a lab mouse, the duration of life for this mouse is about 2 years. So it's pretty much half of the lifetime of the mouse.
Randall, you look like you're dying to ask a question.
I want to know a little bit more about you guys. What's your founding story? How'd you find each other? 30-second version.
Sure. So we met back in 2004 through mutual contacts in Colorado. Dr. Sandler was driving through on his cross-country move, and we kept in touch. Then when I went to Harvard Business School in 2006, and he was working at Harvard, we collaborated on a project which actually resulted in a patent filed. And then 2 years ago, when I moved to New York City, I also reconnected with Dr. Sandler and his family, and we remained in touch and decided to work on HemoGenyx together.
And the other Sandler, presumably, is your daughter? Or
Is my wife.
Your wife. Okay, great. It's amazing, Jordy, how many families you've found.
Yeah, we're all one big family here. It's Buffalo. Bijan.
Yeah, pardon me. Are there other applications for this approach? I mean, it just seems very innovative, and I'm just wondering, does it have other, diseases that it can improve patients' lives besides the ones that we've talked about today?
Absolutely. So we actually plan to begin working on those when we're wrapping up phase 2 trials for the initial applications. There's very promising results pointing to potentially improved treatments for HIV, for multiple myeloma, for sepsis, and for others.
And does that, I guess one of the reasons I was asking, does that help with some of the FDA approvals and clinical trials? Is there any kind of way to make progress fast? I'm just trying to think about how you finance a business like this.
Definitely. So I'm really glad you bring that up. The orphan drug exception that we're able to get, that is because this is directly applicable to a condition called severe aplastic anemia, which is basically bone marrow failure. And the way to treat that is also with bone marrow transplants. So there are very few patients that are affected by this every year. Several hundred, and because of that, we're able to accelerate through clinical trials.
I have a stupid question. How come New York Hospital just doesn't give you the money and become your full partner?
So the hospital actually did give us some money. We were just awarded the Weill Cornell Award for Innovation. So there's definitely some funding coming from there, and you can speak more to the details of that.
Yeah, I just got a Daedalus Award for innovation, $100K from Cornell to do specifically this type of research.
Right.
If you win the top prize, so you're here in Buffalo for at least a year, where is your business after the year end? Are you in phase 2? Are you almost done with your clinical trials? How far along in the process will you go to market?
Yes, so in 1 year, our goal is to complete preclinical trials and go into phase 1 clinical trials with the FDA. And at that point, you can speak to more of that, but the lab facilities will be here. The company will be headquartered here. So we hope to continue to be able to leverage that.
Fantastic.
One more?
One
That's
more.
It? Okay. I'd like to, your thinking on what would be the potential mechanism for this being effective for HIV or sepsis? I don't understand.
Berlin patient CCR5 receptor. So you can take these cells out of the patient, modulate the receptor like people do with CAR technology, and put them back so that the blood cells that are generated from these progenitors will not be susceptible to the infection of HIV. It's basically CCR5 receptor mutation.
Okay. All right, thank you.
Vladislav, Nina, great job. Thank you very much.
Thank you.
Tough questions. You ended them great. Thank you very much for coming.
Appreciate all
Take
the
care.
questions. Thank you.